students. So today we are going to talk all about your next lecture. Next lecture of fundamentals of plant breeding, that is all about introduction. When I say introduction, you or I don't have to introduce yourself or myself. Introduction means here we will be talking for the plant introduction. Then the second thing we will talk about acclimatization, and at the end we can talk about domestication, right? So these three are the topics of your today's lecture, that is plant introduction, acclimatization and domestication. So these all are the contents that we'll be discussing right now. Within the plant introduction, we will talk about what is meant by introduction, like what is the concept behind plant introduction. Then we will also talk about its types. Then see, anything we do, we must be very clear of its objective. If any topic is being taught, then one must be clear. Like what is the objective behind that topic? For example, even if I don't make you familiar with the objective, you yourself should answer yourself at the end. Like what was the advantage of this lecture? That means the concept should be clear to you all. Although we conclude any topic by giving its merits, by giving its demerits, that itself is objective, right? But one must know, if we are learning any topic within any subject, what is the object behind that coverage of that particular topic, that must be clear to all. So here also within the plant introduction, we will talk about the objectives. Then we will talk about the various agencies which are involved in the plant introduction. Then how plant introduction is possible, that is we are going to talk about its procedure, then purpose, important achievements, merits and demerits. Thereafter we will talk for the acclimatization, and then we will talk about the domestication, and these three things we will cover under domestication. One is the selection criteria, then the changes which occur as a result of domestication, and we will often talk about some certain important changes that have already took place as a result of domestication. So very first is the plant introduction. So before I actually go for letting you know about all these statements that I have written, my focus is not to introduce you all to these statements. My focus is to introduce you all with the concept of plant introduction. See, where till the name itself is indicating plant introduction. Here again I repeat, we are not talking for the introduction. Say you should not expect like ma'am is going to give her introduction, like her name is so, she has done C, so and so and so. No, today I will talk for the introduction, but here I am talking for the introduction in terms of plant. And when we talk for the plant, your subject name is fundamentals of plant breeding. So we will be talking all about genetic content only. Say we are talking about genetic content, or even I can say I'm talking about the genotype, right? So plant introduction is what? That is something is being introduced, but what is that something? That something is genotype. And introduce means if this is the plant present at a certain area, if I have to introduce this plant, that means it has to be picked from its particular location, and then it is to be transferred to the other location. That means to the other area. And when it is transferred from one area to the other area, that is what we call as a plant introduction. And very important, the area into which it is to be introduced, it should be the one that no more meets the characteristics of the area into which it was already present. So this is what said to be plant introduction, right? So if I come to the definition of the plant introduction, I hope it is very clear to you all now that in plant introduction is actually taking a genotype or a group of genotypes to a new place. To a new place or to the environment. When we talk for the place, obviously place is surrounded by the environment. So taking the genotype, either a single genotype or either a group of the genotypes to the area, to the place, to the newer area where it was not grown before, that itself is said to be plant introduction. What actually is involved within the plant introduction? That means introduction is very clear. We are introducing new varieties of a crop. Those new varieties which were already grown in a particular area, but we are, see, it is not always like you are to go for a collection of the new, you are to go for the collection of the new genotype and then only it is to be introduced into the newer area. No. It can be either, it can involve either the introduction from wild to the another area, or already this one variety that is already grown to this area, it can also be transferred to the another area. So it involves the new varieties which are grown, already grown. It can involve wild varieties also, or a totally new crop species. I mean to say, here when we are talking for area, basically we talk about the wild varieties. And this wild variety is collected from its area to which it is very much adapted, and then it is allowed to grow under the other area into which it is no more adapted, right? Into which it was never grown before. Basically, we talk for the wild, we basically talk for the introduction of the wild content only. That means wild genotype only. But here, this is the new variety. This new variety can also be picked from this area and it can be transferred to the another area, or a totally new crop species can also be considered. For example, see introduction of rice varieties, introduction of sunflower, introduction of oil palm. Oil palm introduction is done into the Tamil Nadu. And when we talk for the introduction of sunflower wild species, these are done from the Russia, right? So this is all about the plant introduction. When we talk for the types of the plant introduction, then plant introductions are basically categorized into the two broad categories. That is, the very first one is primary introduction and the second one is the secondary introduction. So the categorization is very clear and simple. That is primary and secondary introduction. So here we will talk for the features of primary and secondary, and even we will talk about its differences also. See, two things basically happens. Say this is our wild variety. This is our wild variety. When this wild variety, concept behind introduction is this wild variety is to be picked from its natural habitat and then it is to be introduced into the newer habitat. This one is its natural habitat, and from the natural it is transferred to the newer habitat. If on the transfer of a wild variety from the natural to the newer habitat, if this variety gets well adapted to this new habitat and is released for the purpose of commercial cultivation without any alteration in its genotype, then this constitutes the primary introduction. I again repeat, we are picking up a wild variety from its natural habitat and we are introducing it to the newer habitat, right? Naturally, what happens whenever any variety is allowed to grow under the newer conditions, normally it takes times to get adapted to this environment, and sometimes results can be like it is no more comfortable in adapting itself to the newer environment. But if I talk for the first prospect, that means where it is comfortable in adapting itself to the newer environment, then it will grow. It will grow. Now, what is our motto in the plant breeding is to make a variety available so that it could be commercially grown, right? If it is growing into the natural habitat, and conditions when it is growing into the natural habitat is very important, there is no change in its genetic content, because here the conditions are newer conditions. So normally what we expect, like whenever this wild variety will be allowed to grow into the newer habitat, there are going to be certain changes in the characteristics of the genotype. But normally this is what we normally expect. But it is not like whenever a wild variety will be allowed to grow into the newer environment, all the time its genotype is going to be changed. No. But when we talk for the scenario where genotype is not changing, there we can release this particular variety, and the release is with the motto that it could be commercially cultivated. This process where the genotype easily adapt itself to the newer environment and there is no genotypic change in the variety, then we can commercially release this particular variety, and this is what that is constituting the primary introduction, right? But it is very less common. As I already said, whenever we will be going for the change, when we will talk or when we will focus for the change of environmental conditions, then for sure 100% chances are there, like there will be change in the genotypic content, because the wild variety is always adjusts itself to the natural conditions only. Whenever there will be existence of the variations, perfectly there is going to be change in the genotype, right? But here we just talked about that, like there is no alteration in the genotype, and this is only the particular reason we are looking after its multiplication, right? So this is normally less common. This kind of scenario does not happen regularly. It is less common, right? And these all are the varieties which are introduced from the different countries into the different countries. Now we have to talk about the secondary introduction. In the secondary introduction, what is happening out, this thing is done. Variety is picked up from its wild habitat, and it is allowed to grow into a newer habitat. Okay, it is getting adapted itself to the newer environment. But now, before it is released for the commercial cultivation, here one aspect is to be considered, and that aspect is said to be selection. That means here we didn't go for any trial and directly we released the variety, because we were knowing there is no change in there is no alteration in the genetic content. But here when we talk for the secondary introduction, that means here we need to work on a prospect that is said to be the selection, and further this variety that is introduced further into the newer environment, it can be used in the hybridization program so that a superior variety could be developed. That means after introduction, primary aspect is what? Introduction. After the primary introduction, we are going for the selection to see, like the genotypes or the features we were looking after are exactly the same. They are present or not. If they are present, then those particular varieties are selected, and further they are used in the hybridization program so that they can be used for the development of superior variety. I will explain it with an example. Say the variety that is transferred, say the variety that we have transferred here, it is a feature, it has a feature that gives, it has a feature that gives higher yield. So it is introduced. After introduction, it will adapt itself to the newer environment. Then it is selected. Selection is always done on the basis of certain parameters. So it is selected on the basis of certain parameters. And after the selection, say that variety is S1 that is selected. We know this is the variety that is giving the higher yield. That means it is having somewhere the feature of your desire. We know that all the time it is going to grow, all the time it is going to give higher yield. This is said to be selected variety. Now, what we always want like the yield should be good. Even the varieties which are not important in the giving the higher yield, they should also give the higher yield. So what we do, we cross this selected variety with the other variety that is no more giving the higher yield. And the cross is done with a motto that it's because we know some of the genes from one parent and some of the genes from another parent, they in turn come into the hybrid, right? So if our one parent is having good features, if it is allowed to cross with the other parent that is not having the good feature, then what happens? Some of the genes will come from this parent and some will come from this parent, and as a result, we will have the hybrid that is giving you more and more, or say higher yield. So the introduction, where the introduced variety is further subjected to selection, and later on even used in the, this is said to be hybridization program, and even used in the hybridization program to develop a superior variety. This actually constitutes the secondary introduction. That means very first step is what? You just change the environment of the wild variety. Once you change the environment, this is said to be primary introduction. But after the changes, you introduce this variety to the selection, considering the certain parameters. And if the variety is selected, it will be further introduced into the hybridization program, just with a motto for the development of a hybrid variety. This is more common, right? And its examples are like in soybean, EC39821 was introduced from Taiwan. This variety existed in Taiwan, and from Taiwan it was subjected to selection, and a newer variety called as variety Co1 was developed. Similarly, when we talk for the rice, in the rice AST4, it was crossed with IR20 to get, this is again the hybridization program. So these are the varieties which are introduced from certain another reason, but after the selection it is giving rise to a newer variety. Here that newer variety is CO1, and here the newer, that is superior variety is CO44. So this is all about primary and secondary introduction. Now objectives of the plant introduction. Objective must be clear to you all till now, since we have covered the basic concept of plant introduction. So the concept is what? Objectives are what? If we are going to have newer and newer varieties, and if say all these newer and newer varieties are good in terms of genotype, then what we will do with these varieties? We will look after its multiplication. And multiplication is not to be done with hand. Multiplication is not to be done in talks. Multiplication can be done by utilizing certain instruments, and those instruments are all present in the newer industries. That means we can build up new industries also. New industries can be built up. For example, because we are to introduce the new plant species, then if the new plant species are to be developed, obviously we are to build up new industry. For example, in case of oil palm. Second objective of the plant introduction is to increase the yield. Third is we used to have a gene pool. More will be the development of newer and newer varieties. More wider will be the area of genotype. That means it is also going to enrich the germplasm collection. We can get the new sources of resistance also against a kind of biotic and abiotic stresses. Like we can develop the basal rice variety that is no more susceptible to the saline conditions, right? And ornamentals can be introduced for the aesthetic values also. Here we will talk about the agencies which are playing a major role at the time of plant introduction. But before we talk for the existing introduction, I would also like to inform you, like most of the introduction, they occurred very early in the history. And in earlier days, the agencies that were playing a major role in the introduction were named as invaders, travelers, traders, explorers, pilgrims. And if I give a name of the one agency, that is naturalist Muslim invaders, then they introduced in these were the agency. This was the agency that introduced in India cherries and grapes. Cherries and grapes. If we talk for the another like Portuguese introduction, then they introduced these all varieties. East India Company brought tea. Botanic gardens played a major role in plant introduction. These are the agencies. And because many agencies were existing at a different level, so a centralized agency was initiated in 1946 at IAR, New Delhi. Then in the 1976, National Bureau of Plant Genetic Resources was started, and the responsibility of this bureau was to introduce, and the germplasm that is introduced into the newer area, its responsibility was to introduce and then maintain that particular germplasm. There also been opening of an plant introduction organization in Dehradun that is said to be Forest Research Institute, and this institute also has the responsibilities like introduction, then its maintenance, and it was also going for conducting various types of tests of germplasm of forest trees. Then once at a central level, agencies were developed. Then at the national level, when the industries were developed, then when the forest at certain states, forests were developed, then they even developed the central research institutes. And the motto of developing this central research institute was to maintain the working germplasm, because it is all about research institutes, and research could be continued only if we have a germplasm onto which it would be worked out. So it was mainly focusing on the maintenance of working germplasm. Again, I repeat, it focuses on the maintenance of a working germplasm, right? Working, because research could be conducted only if there is a certain base present to the researcher, right? If we talk for the India itself, then in India, see students, whatever introduction we talk about, whether we are talking about the introduction from other country, from within the country, from state to other state, any introduction when we are talking about, then it must be very clear, any introduction, whatever route it is we are considering for its introduction, but all those routes are to be, whether we are following this route of introduction, we are following this route of introduction, or either we are following this route of introduction. But remember one thing, all the introductions, all the routes of introduction, they are to be passed through NBPGR. That means any introduction, if it is taking place, it must be in the knowledge of NBPGR, which is the National Bureau of Plant Genetic Resources, right? This is the National Bureau of Plant Genetic Resources, and its responsibility is to look after what kind of introduction is being done. And if that introduction is significant, then introduction means what? Again I repeat, we are doing the introduction of genotype, right? We are going for the introduction of genotype. Then after the introduction, this genotype must not be lost. So before it actually lost, we are this is often the responsibility of this bureau to maintain this genotype after the introduction. So whatever channel of whatever route of transfer or say introduction we follow, all those routes are to be passed from the, it has to be brought into the knowledge of NBPGR, right? So all the introductions in the India, they must be routed through NBPGR, New Delhi. Bureau function, I have already told you, maintenance and introduction. And further it acts as a central agency. Central agency, it is acting just for the export and introduction of the germplasm only. But when we talk for the international levels, this is what at the national level, any introduction is looked after by NBPGR. But when we talk for the international, then it is being looked at by IBPGR. Now procedure of plant introduction. How we can go for the plant introduction? There are to be certain steps which are to be followed for introducing a variety from one area to the newer area, right? And these all are the steps that must be followed. Very first, you need to submit the requirements, what actually kind of germplasm or genotype you require. Then you need to give the address, like it is available here and where it is actually to be delivered. Then once the address and all the details are submitted to NBPGR, because I said all the introductions are to have to follow a particular route, and that route is like all the introductions are to be brought into the knowledge of NBPGR. So NBPGR will look after the after receiving the address and all the requirements, it will look after whether the introduction is taking place at national level or whether it is taking place at international level. If it is taking place at national level, it will hand over the content to the next one. And if it is taking place at the international level, then it will first bring the matter into the knowledge of IBPGR, right? Then the material will be issued and it will be sent to that particular address. So here are the details of how plant introduction can be done. So any scientist or any will first submit the requirement to the NBPGR, because if you want something, you needed to bring that particular thing you need, you have to brought that into the matter, into the knowledge of higher authority. And here the higher authority is National Bureau of Plant Genetic Resources, right? So scientist or university or whosoever needs a requirement of germplasm, that will submit the requirement, what kind of germplasm, how much amount, and all those requirements details, that will be submitted to the NBPGR, that is National Bureau Plant Genetic Resources. Then the National Bureau Plant Genetic Resources, after receiving the information or say the requirement, it will look after the introduction is to be done within the country, or introduction is to be done at the international platform. If the introduction is to be done from other country, that is at the international level, then NBPGR provide that address. That means it will say, it will request the International Bureau Plant Genetic Resources to give the effective supply. IBPGR will come into domain and it will work, and after working, when it will assign task to the further, and after the collection of the material from the source, it will quarantine them and will pack them. Quarantine is done to see, like along with that along with the introduction of the genotype, there is no more introduction of any diseases, etc. Then it issues certificate based on the material, and finally it will hand over that particular material to NBPGR only. Because when the level comes, when one has to work for the introduction from other countries, very important, from other countries, then the major role is played by IBPGR. But after playing the major role by the IPGR, it will be, the task will be again handed over to the NBPGR only. Because I said whatever the route of introduction is being implemented, all those routes are to be brought into the knowledge of NBPGR, that is National Bureau Plant Genetic Resources, right? And at the end, it will be sent to the NBPGR, right? And NBPGR since had the requirements and it had the address, so it will transfer it to the particular address that will be mentioned in the requirements. So this is all about procedure that is followed out in plant introduction. Next is we will talk for certain more roles of NBPGR. Like I said, it assign number to the material. Like whatever material it has obtained, say if this is the material that is to be transferred at a particular address, very first task or responsibility of NBPGR is to assign number to this material. Say the number that is assigned is 1. Whole of the material will not be issued or it will not be transferred to the particular address that would be mentioned in the address. It would not be, all the material will not be transferred to this particular address that is mentioned by the man, say that is university or say that is any scientist. Part of it is kept for the, part of this is kept for germplasm. And the second part, half part they retain with themselves, and the other part is only the part or is only the portion that is handed over to the scientist. But it is not like all the requirements will be fulfilled. No, there are certain restrictions on the certain requirements. Like if say someone means, say if university or the researcher asks to transfer or introduce Nidran banana, then its introduction is totally restricted. The reason behind the restriction is like it is actually this Nadran banana is the one that is actually found in the Tamil Nadu, and it will not be sent out of the Tamil Nadu because this banana is associated with an occurrence of the disease that is called as bunchy top disease. So if NBPGR don't restrict this introduction, then along with the transfer of the Nidran banana, there will also be the transfer of the disease that is said to be the bunchy top disease. So any introductions which are further combined with certain kind of diseases, their introduction is totally inhibited, their introduction is totally restricted, right? Similarly, we cannot import cocoa from Africa and West Indies, sugarcane from Australia, sunflower from Argentina. Just the reason is very simple, like these all are associated with the diseases, and if we don't restrict their introduction, then it will also carry the diseases along with it, right? Here are the certain functions of NBPGR that I told you. Very first is it is looking after the introduction, that is transfer of the genotype from one area to the totally to the entirely newer area. After the transfer, it will look like the newer genotype is maintained and it is no more disrupted. Then it is distributed also. It provides the information about the germplasm, like and you know all this information is not what is the mode of providing the information, like whatever information they used to have about the germplasm, they publish it into the publications, right, into the research papers. They even conduct various training courses for the scientists, so that they come to know how introduction of the germplasm is to done, and how after the introduction the germplasm within the newer area is to be maintained. They even conduct exploratory surveys because germplasm they have to provide the requirement of the university or say the researcher. But how they will fulfill the requirements of the university or the researcher? They will fulfill the requirements if they have content with them. I say for example, just considering an fake example, I ask Gulshan to provide me one notebook that is having all the notes of your fundamentals of plant breeding, right? But before Gulshan actually gives me all the notes of fundamentals of plant breeding, including the topics which I have not covered, for that he needs to sit and he needs to work hard so that he could prepare all the notes within the notebook, and then he will hand over that all the notes to me, right? In the similar cases, scientists or university, they are they are for submitting their requirements to the NBPGR. How this NBPGR is going to fulfill the requirement of the scientist or the university? If it is having content with it, then only it will transfer further, no? So so for providing the germplasm to the university or to the scientist, it carries many surveys, and those all surveys are carried just for the collection of germplasm. They also go for setting up of natural gene sanctuaries. These all are the functions of NBPGR. But if we talk for the purpose of plant introduction, then it is very clear till now, like it is looking after the introduction of entirely new crops. All those new crops are used in the scientific studies with regard to study on biosystematics, evolution, and origin of plant species. And you know, certain varieties which are introduced are also used for the aesthetic value, that is for the ornamental purposes, for the beautification, etc. For example, we talk for the ornamental shrubs, lawn grasses, they all are introduced just to satisfy the finer sensibilities of the human. Often they are of a great value in terms of decoration, and even they play a very important role in the social life. Newer varieties can be developed, like if we talk for the plant introduction, that means if we talk for the primary plant introduction, if we talk for the primary introduction, there is what happening out. The concept behind the primary introduction, there is no change in the genotype. And since there was no change in the genotype, that was only the reason it was directly being released, and even it was multiplied commercially. So new varieties can be released as a result of plant introduction, and varieties could also be saved from diseases or pests. So this is the purpose. These are the major achievements we have received till now, new crop species are evoluted, varieties are directly released for commercial cultivation, varieties are selected from introduction, and these are further used in the hybridization to develop the superior varieties. Develop the superior varieties. This I have discussed in say secondary introduction, and this is a part of primary introduction. So these all are the achievements of plant introduction. So here are the merits, that is the advantages of plant introduction. Very first advantage is it is providing new crop varieties. If it is providing the new crop varieties which are giving higher yield and can be directly used for the commercial cultivation, that means it is providing the new plant species also. It often provides the parent material. Parent material, as I told you, the case of hybridization. In hybridization, I said after the primary introduction, the variety is selected. Selection is carried out in regard to certain parameters. Once the selected variety is of a good characteristic, it is allowed to cross. Say that selected variety is one. It is allowed to cross with the other that is no more giving the desired results. So as a result, by crossing this with this, even its characteristics are improved. So this is what Enrichment, this is what? It provides the parent material for genetic improvement of economic crops. Then there is the enrichment of germplasm. It increases the variability also, because the more and more newer genotype is being added up into the gene pool, more are the variations that are existing. And it is the introduction is also protecting from the transfer of the diseases. So these all are the merits of plant introduction. But there are certain disadvantages also, and those disadvantages are like, otherwise NBPGR is properly monitoring the introduction aspect. But if unfortunately somewhere certain things are missed, then it may involve the introduction of new weed, but unknowingly, right? Secondly, again I repeat, likewise NBPGR is monitoring all the activities which are associated with the introduction. And monitoring is done very carefully. But unknowingly or unfortunately, it may lead to the introduction of new weed, or it may lead into the introduction of new diseases. For example, even after being so much careful, it could be observed like late blight of potato. This is the disease that is found in the potato. From Europe, it was like evolved. It evolved from the Europe. And Bunchitop, I have already told you. Then new pests can also be transferred along with the introduction. Sometimes the one that we considered as an ornamental, they became weeds. And because what we are doing, we are transferring one genotype from its natural habitat to the another newer area. It is actually resulting into the ecological imbalance, because earlier its amount was perfect in this area, but once certain of the genotype is transferred to the another area, here the content, the amount of this genotype decreased, and here it increased. So it also result into the imbalance of the ecology, right? Normally what we say, ecology must be balanced. But in such scenarios, in such cases, as is the case of plant introduction, here occurs a certain imbalances, and those imbalances are said to be ecological imbalances. So this was all about say plant introduction. Now I will be talking about the next topic, that is acclimatization. But before I discuss the acclimatization, I would like to make you familiar with the domestication. Domestication is what? Here also we are changing the environment, but here wild species, they are taken from their natural habitat, and then they are allowed to grow under the human skills. Very important. Domestication, that means we are talking for domestic things. Domestic is what? When we say certain घरेलू aspect, that is said to be domestication. And human resides into the say homes. So here we say like whenever any wild species is allowed to grow under human management, this process where wild species are brought from their natural habitat to the under the human management, then it is said to be domestication. That is the process of bringing, again I focus, process of bringing wild species, process of bringing wild species, very important, wild species are brought from their natural habitat, and they are allowed to grow under human management, right? from wild species are brought from their natural habitat and allowed to grow under human management. If we talk for the present day cultivated plants, they all had been derived from wild weed species. Whatever you are growing nowadays, whatever is being grown, that all is the result of domestication, that is the result of human management. Most of the crops had already been domesticated by the prehistoric man, means the those who were present in the before us. And under domestication, what is done? When any crop is picked up from its natural habitat and it is allowed to grow under the human management, then certain changes takes place, and these changes are totally different from the species from which they originated. Actually, I give you one example. This is one wild species, and when then this wild species was allowed to grow under the human management, then a newer variety evolved. A newer variety evolved. A newer variety is evolved, and the features of this newer features of this newer variety are no more similar with the variety from which it actually originated, right? That means both are having different features. Say it was of red color and it turned into yellow color. Just giving a fake example. That means once under the human management, then human will allow this plant to grow according to its desire. And as a result, there are total genotypic changes, and as a result of those total all genotypic changes, there emerges a new variety that no more resembles the variety from which it originated. So, as a result, the changes which are taking place, they are again selected. Again, selection is done because it is no more resembling with the parent from where it has actually evolved. So we need to test these features. So these features are selected by man, because see, man can control this plant, but man cannot control the environment. That means two kinds of selections are going to take place. When one selection is being done by the human, then natural selection will also take place. That means natural and artificial selection both will take place, and the variety that is developed as a result of domestication will be called as distinct species, because it entirely differs from the features of this one, that means from its parents. So this will be called as a distinct species, right? How selection can be done under domestication? How selection can be done? See, if we talk for the population, then in the population we have number of individuals. And any individual can mate with any other individual independently. Before I actually talk how selection is taking place, I made you all familiar with the term population. And I said population is the group of individuals, and within that group, the individuals which are present, they can mate freely with each other. So, because within the population, individuals can freely mate with each other, so the selection is basically divided into two categories. Very first is natural selection, and the other is called as artificial selection. We will talk one by one about those selections. This is natural selection. Natural selection, name itself is indicating that is a selection that is taking place due to the natural forces. And when we talk for the natural parameters, then the natural parameter very first is the soil, then climate, and other factors of the environment. And if we talk for certain biological factors, then it includes diseases, insects, pests, etc. Right? Natural selection will only occur in natural populations only. And natural populations are the one that are wild forms and its related species, right? And all the genotypes of this population are going to reproduce. The one that becomes adapted to the existing environment, they will be having the genetic variability among them. This is a natural selection. That means a selection that is taking place under natural parameters. And artificial is what? Where man interference is involved. That means the selection that is carried out by the man is said to be artificial selection. It is basically confined to domesticated species only. And here, like in the natural population, random mating was taking place. Here only selected plants are allowed to reproduce. That means within the population, any individual, that means any plant cannot mate with any plant randomly. Here, say here are the 4 species, then because man is controlling this kind of selection, so he will ensure like one kind of plant is allowed to mate with this particular kind of plant only. That means here selected plants are only allowed to reproduce. This artificial selection is the criteria that is giving more useful results to the man. It leads to the marked decline in genetic variability plants. But here, because if the natural mating is taking place, then the naturally they will get adapted to the natural environmental conditions. But here, because man is interfering with the genotype of the content, so here the adaptations to the natural environment are less. And we are to provide carefully. We need to provide very managed conditions so that these crows could grow. Most of the present day crops, these are the result of continued artificial selection only. So there are certain changes that takes place in plant species when plants are allowed to grow under domestic conditions. All the characteristics will be affected. This is the very important concept that everyone must know. That like all the characteristics, they will be affected under domestication. But the characters that show more changes, see all the characters are going to be affected. Say that character is related to the height. Say that character is related to yield. Say that character is related to the disease resistant feature. Or say it is related to the pest resistant. Any character is going to be affected by domestication. These are only the 4 characters that I discussed, but as you all know, there are number of features of the plant. So whatever feature of the plant is available within a specific plant, all those features are going to be affected under the influence of domestication. But where the more like we said, no, when domestication is taking place, then as a result we have entirely new plant. Where I shown you one example where we used to have red plant and then a totally newer variety emerged that was giving yellow color. That this newly developed, which was no more meeting the features of its parent also, this was said to be distinct variety. Now my point is, what characteristics, like say we had 4 characteristics in the plant, out of these 4 characters, which are going to be the distinct characteristics? Very important. But the distinct characters are going to be the one onto which selection is carried out, because I told you selection is to be carried out in respect to certain parameters. Say the distinct changes, that means the totally different genotype will be produced only in the cases where certain features being the object of selection, because when we are carrying out selection procedure, we need to focus on certain parameters. That means we need to define the parameters on the basis of which selection of a particular variety is to be done, right? So where the distinct changes are going to come is basically dependent upon the objects of selection, that is the criteria of selection, and are still plant breeding objectives in many cultivated species. One is we have already decided certain parameters, and another certain parameters are still evolving on the basis of which domestication could be done. So these are the changes that occurs in the plant species, and as a result we have entirely new species, that different species that is said to be distinct species. Here I will talk about the certain changes that have occurred, like elimination or reduction in shattering, elimination of dormancy, decrease in toxic content or undesirable substances, and obviously plant has been extensively modified, right? So these are the some of the changes that have taken place. Changes are more in number. Decrease in plant height could also be done as a result of domestication. Increase in height, if we can look after the decrease in height, then obviously we can look after the increase in height also. Life cycle can be duration of the life cycle can be shortened. There can be implementation of increase in the size of the grains, and we can also look after the increase in economic yield. Asexual reproduction has been promoted because man, when we talk for the artificial selection, then in the artificial selection, selected processes are being done. That means those selected processes may involve asexual reproduction also. So here, what kind of reproduction is promoted is the asexual reproduction. Polyploidy is achieved. Even we can look after the change in the sex form of the species, like dioecious, bisexual forms. They all are developed under domestication only. And in certain cases, self-incompatibility phenomenon has been removed. Variability within a variety has drastically decreased. Then this is all about the changes that takes place as a result of domestication. Now, introduction is clear to you all, domestication is clear to you all. Now, I would like to make you familiar with a term, that is acclimatization. See, till now we talked about the concept. Very first we said plant introduction. And within the plant introduction, we transferred a genotype from its natural habitat, that is wild species we took, and we transferred it into the newer area. This is introduction. Then we talked about domestication. And in domestication, we said here also introduction is only done, but the introduction is under the human management. Introduction is under the human management. Now I will talk about acclimatization. See, when any varieties, when any superior, why you are picking this variety, this wild species? Obviously because it is superior. That is only the reason you are selecting this variety, and later on you are looking for its management under the human observation, or then you are transferring it to the newer area, right? Normally, as I said, in the case of primary introduction, which takes place very rate of which is existence of which is often, in that case, as I said, no, normally what I said in the primary introduction, this is capable of adapting itself to the newer environment, and there is very important, there is no change in the genotype. There is no change in the genotype. And because genotype was not changed, it remained same as of its parent. This was the reason it was commercially cultivated. It was released as a variety for commercial cultivation. But on the other hand, I said this is the rare phenomenon that takes place, because whenever anyone is allowed, even when I talk for you, when you are coming into the hostels and you are living, you take a bit time to adapt yourselves to the hostel environment, right? In the similar way, this is the natural concept, like whenever any superior variety is going to be transferred from area of its natural habitat to the newer area, there are going to be certain changes. But yeah, it is also possible, like with the time it will adapt itself to the newer environment, because it is a very important principle of the biology, like those who will adapt themselves to the existing conditions will survive and others all will die. So if you want yourself to survive, you have to, if you want yourself to complete the BSc honors degree, and if you are staying in the hostel, so it is very common, like you have to adapt yourself to the environmental conditions of say college and of hostel, right? So when the superior cultivars are taken from the distant region or say the neighboring region, and when they are introduced into the newer area, then normally what happens, they initially fail to produce a phenotypic expression as they used to place in their natural habitat. When you see, I will implement this example onto you all. When you were say many of the students of you people are from Bihar, right? Some are from Punjab or other states. When you were studying in the Bihar, you were obtaining very good marks, right? But when you came into the Punjab, your environmental conditions totally changed. Even the teaching methodology, technologies, many things you must have felt like they are changed. So in the initial period, you must have observed, like in the first semester, many of you didn't get good marks in the first semester as you got in the 12th examination that you cleared in the Bihar. The reason was that you could not adapt yourselves to the conditions. Initially, you failed to survive, you failed to produce the same result as you were giving in the Bihar, right? So, whenever any superior variety is introduced into the newer area, initially, again I say, in the starting stages, it fails to produce phenotypic expression, very important phenotypic expression, those phenotypic expression which it was giving in their place of origin, right? But it is not like all the time you are going to get good, the less marks only. In the second semester, what happened? You adapted yourselves to the teaching technologies existing in the Amritsar group of colleges, and then you achieved more better results. That means now you are giving the results same as you are giving the same results in Amritsar group of colleges as you were giving in your 10th examination that was conducted in the Bihar. That means initially you were failing to produce the same results. Here results are phenotypic expressions, but later on you picked up all the environmental conditions, and here if I say plant picked up all the environmental conditions and it gave the optimal phenotypic performance. This what we call this concept behind this adaptation that took place into the newer environment, and as a result of adaptation, when you are giving the same results which were being given in the Bihar, this is actually said to be acclimatization. And in short, I would say adapting yourself adapting yourself to the newer environmental condition, to the new environmental condition, is all said to be acclimatization, right? So this is said to be acclimatization. This was a flow chart that I just prepared for you all to understand the process of acclimatization. So definition of acclimatization is very clear, that is ability of the crop variety. Here I won't be talking about you people, I am just focusing on crop varieties. So the ability, very important, ability of the crop variety, that it is becoming adapted to the new climatic and edaphic conditions. Edaphic means the conditions which are related to the soil. Once the crop variety becomes adapted, to the newer climatic and edaphic conditions, it is said to be acclimatization. So here actually how you are able to adapt yourself, because you adapted to the new technology. You, once the lectures were delivered to you, you went for more and more revision. You try to catch the concept that is being delivered to you all. So in the similar way, crop varieties could adapt itself to the newer environment because it followed an increase in frequency of those genotypes. Because see, there used to be see, if I come back to you all, there used to be two concepts. One is you improve yourself in the second semester. That means you give the same results as you were giving in the plus two examination that were conducted in Bihar. But it is not like all of you gave the good results that were same as this examination. Many of you didn't adapt yourself at this stage. And now you are looking after your adaptation in the third semester. I gave one example, like immediately you are adapting yourself. How you will adapt yourself immediately in the second semester, like here you will more focus, your vision is going to be more clear, and you know, like in this semester I have to obtain all those objectives that I could not obtain in the first semester. That means your criteria is very clear. And while you were here and while you came into the AGC, you also recognized the certain problems due to which you were unable to obtain the same results, right? That means you focused on these frequencies, you focused on the frequencies of these problems, and you slowly and slowly you modified, you improved these frequencies, and these problems later on turned into the success. And then you could give the same result in the second semester as you were giving in the plus two examination that were conducted in the Bihar. This is just a basic example. You need not to mention this example into your written up content. This is just for your understanding, like what I mean to explain with the acclimatization, and that I'm trying to clear with giving you a very basic example that is one of the part of your daily life, right? This is a concept of study where you were focusing on the problems and then you converted the frequency of these problems into the higher and higher frequency of the success. But here, when we are talking about plant breeding, here frequency of those genotypes. Here also within the plant there are two kinds of genotypes. One say we will improve and other say we are not going to improve. We need not to give the better result. But here adaptation within the crop species is focusing on increasing the frequency of those genotypes which could adapt itself better to the newer conditions. That means they neglected their this part and they just focused on to this part, and as a result they got better adapted to the newer environment. But finally, this is very important, like success of acclimatization of course is controlled by the two parameters. Very first, the place is having very much effect, because see in the second semester some could not give the better results because maybe their company was not good, maybe they were not staying at a good places. Means there are number of the parameters which inhibited their conversion of problems into the success. So place is playing very important role, and in the adaptation to the newer environment. And secondly, you yourself need to decide whether you are to improve or whether you have to again come to the lower standard, right? uh, so this is the selection of new genotypes. You need whether if because see if the plant is going to focus on these kinds of genotype, then it would have never adapted itself to the newer environment. It could adapt itself to the newer environment because it focused on the genotypes frequency of those genotypes which were about to give its a better adaptability to the newer environment. So these two are the factors which play very important role in the acclimatization. So this was all about your today's lecture. That means this was all about say plant introduction, domestication and acclimatization, right? And with this I complete my today's lecture. If you have any query, any doubt, you can directly contact to me. So thank you for patiently listening to me. Thank you. Stay home and stay blessed.